Well, I'm going to still start with our forward-looking statements. We are a publicly traded company, and I may be making some forward-looking statements. You can refer to our safe harbor and risk factors through our filings. I'll be the only speaker today, and I'm really pleased to be here. This is a little bit of a different forum for us. Lineage Cell Therapeutics probably fits in very nicely with this third annual conference, but you may not know a lot about the company. But one of the ways in which I think we're different is that we are really progressing nicely with some of the promises of regenerative medicine and moving them forward into human studies and generating data from those studies. So our approach is quite straightforward. We transplant cells to treat various diseases and conditions. And these are off-the-shelf cells, so we can manufacture tremendous numbers of them. And really, we think of ourselves not so much as cell therapy, but rather as transplant therapy or transplant medicine. We have a world-class manufacturing with wonderful accomplishments that we've made, and I'll describe in scale. We have many hundreds of patent applications, and we have right now 3 active clinical programs. We have something we call OpRegen, which is in a phase 1/2A trial for dry age-related macular degeneration. That's one of the leading causes of blindness. We have OPC1, which has completed a phase 1/2A clinical trial in spinal cord injuries. And we have ACT2, which is our oncology platform, and which also in the clinic currently in non-small cell lung cancer. And one of the things that we find very exciting about cell therapy as a whole is that it may be able to deliver results which are not achievable with traditional small molecules or antibodies and other conventional pharmaceuticals. And in fact, we've seen some evidence of that. Our company, Lineage, has reported the first ever finding of retinal tissue restoration in a dry AMD patient. And I'm going to explain in a few slides exactly what that means. We also have seen incredible benefits in individuals who have suffered from spinal cord injury and their ability to regain mobility after treatment with our cells. And we've seen strong inductions of immune response in patients with cancer. Each of these have very significant commercial potential, and we have some very exciting data still to be released this quarter and next quarter, and frankly throughout the year. And the company is well funded and has been increasingly noticed by the investment community. So Lineage is important in the area because we are trying to bring forward essentially a new branch of medicine based on the transplant of specific cells into the body. We begin with undifferentiated stem cells. Now, I'm sure you're aware that there are a lot of companies that do work with undifferentiated stem cells, or maybe they make some light modifications. We do something very different. We fully differentiate these cells. So we don't actually ever put a stem cell into a human being. A stem cell to us simply represents starting material. It is a cell which has the potential to become any of the cells, the 200 or more cell types in your body. So these stem cells are able to become liver cells or bone cells or brain or hair. They can become anything. So they have that machinery inside of them. And what we do at Lineage is we drive their differentiation. We direct their maturation into specific cell types, and only those cell types. And we don't alter the DNA. We're not manipulating the DNA. You've probably read about some of the concerns that are associated with editing the genome, but we just use, let's call them natural compounds and external forces in order to encourage these cells to become just a single cell type. And we've been able to do that to form retina cells, spinal cord cells, and immune cells. And because these stem cells are essentially infinitely reproducible, we can make huge numbers of them and then convert them into these discrete cell types, which then are used in the clinic. We do this at our clinical facility. We have multiple GMP suites. This is where a lot of our intellectual property is established and where we bank and manufacture our clinical material. These are those 3 clinical programs. I'm quite proud that each of these programs has received significant external support, whether that's from the Israeli Innovation Authority for our Vision program, the California Stem Cell Agency for our spinal cord program, or Cancer Research UK, which has provided more than $10 million of in-kind support for our cancer program. And again, all of these are in the clinic. This is not basic research. These are actual therapeutic candidates moving through human trials today. The most leading program that we have is for dry age-related macular degeneration, a major cause of vision loss. What happens in this situation is, frankly, you're outliving your retina cells. They begin to die off for reasons that are not fully known, but typically late in life, 70s, 80s, even 90 years old, you may begin to lose a special type of cell in the back of your eye called an RPE cell. And because the eye is a window, you can look in through that window and you can see what's going on. And through these 6 small panels here, you can see these black areas which are growing. That is actual visualization of the area of atrophy. Those are retina cells that are dying off. It's almost like a wound or an abscess, which is getting larger. And at the same time, this individual's vision is becoming impaired. They start with normal 20/20 vision back in 2012, and they became legally blind in just a few years, and now all the way out to 26/40 for visual acuity, which is extremely poor. And what's important here is that as these cells die off, this is irreversible. Humans do not have the ability to regenerate this tissue, so you cannot recover from this. It is a progressively, directionally worsening condition. And it is an enormous problem. There are 2 types of AMD in the world, dry and wet. We do have therapies for wet AMD. We do have traditional small molecules and antibodies which can help treat that. And they've been wildly successful, over $10 billion in sales around the world for those therapies. But yet there is nothing approved by the FDA to treat dry AMD. It really invites the question, why? Why is there nothing to treat dry AMD? And we think the problem is that by the time cells are dying off, so many things have gone wrong that you can't affect a single pathway. You can't make a small or minor biological interruption or change to a cell and think you're going to completely intervene in the trajectory of the disease. We think the only way to accomplish this is by replacing the cell in its entirety. This is a cartoon version of what that looks like. On the left, this is a cross-section of a retina. It's a diseased retina. Those orange cells are the RPE cells. Those are the ones that we care about. Those are the ones which we replace. And as they die off, they no longer can support the other cells around them. Those are the photoreceptors, which are critical to your vision. And as they die off, waste accumulates. That's that yellow material called drusen. So what we do is we manufacture outside of the body from those cell lines, we manufacture brand new retina cells, and we are able to transplant those to the eye. And in doing so, we're looking to rescue those photoreceptors. We're looking to clear that waste material. And this is why I've described it as transplant medicine. Our clinical trial recently completed its enrollment. We had 24 patients that participated in the study. The first 12 of them were legally blind, so they had very advanced disease, and we weren't sure that we would be able to do very much for them because they had such advanced disease. But it was an important safety component to treat just one eye in people who had very poor vision at baseline, and then see how well the material was treating, whether the surgical procedure was acceptable, and so forth. And we found a lot of encouraging data. We saw evidence of architectural changes in the retina, clearance of some drusen material, that waste material, and it encouraged us to move into the second phase of the study with patients who had better starting vision. These are patients who had more to work with for us, and they had earlier stage disease. So we thought we may be able to see better outcomes in those patients. And in fact, that's exactly what we did observe. Some of the findings that we reported publicly last November were that these cohort 4 patients, these better baseline patients, some of them had some really striking benefits. That area of atrophy was growing slower. They could see better. They could read faster. The retina structure was improved. The waste material was reduced. And it's important to note that this single administration of our cells, a single transplant of our cells, has in no cases been rejected. We've had 0 cases of either acute or delayed rejection. The cells have been resident and they have been stable for many years. We have patients now out beyond 5 years who have maintained these cells. But probably more important than all of that together is that we announced that we had found one patient who had a phenomenon of retinal restoration, meaning the area of atrophy got smaller. And if you recall, I said that that's impossible. And it is impossible naturally. You cannot heal. Every once in a while, someone manages to shrink a tumor on their own and defeat cancer, and it's wonderful. But that does not happen with dry AMD. You cannot heal this area of your body. So the fact that we were able to find someone who had a smaller area of atrophy and it persisted for 2 years now has been incredibly exciting. And we think that that is something that really affirms that cell therapy may be able to deliver some outsized returns in this area. This is a graph of the data of patients in cohort 4 who received our treatment, and it's a measure of how many letters they could read in an eye chart. Really straightforward. The green line represents the patient's treated eye, and the orange line is the patient's untreated eye. We only treat one eye in these individuals, and you can see that the treated eyes actually increased their ability to read letters on an eye chart compared to what's called the contralateral eye, which was essentially unchanged over the period. Is that important, gaining 10 or 15 letters? Yes, here's what that looks like. This is illustrative of a 10 or 15 letter gain. It's the difference between reading the K or reading the V or the Z in this eye chart. So these are significant changes. I spoke with a woman just a couple of weeks ago who called her experience life-changing, and she only gained 8 letters. So very exciting to be able to have that kind of effect on an individual. We also are very interested in how quickly these areas of atrophy grow, because as they get bigger, they create greater interference with your vision, especially when they hit the macula. And the inversion of these lines now, the lower green line is indicative of slower growth of the atrophy, and the orange line, which is untreated, is faster growth. So this is great. This is exactly what we want to see. We want to see a slower growing atrophy. We want to see improvements in vision. We want to see durable, stable, and safe therapeutic interventions. This is all very exciting for us. These are some overlays, a lot of data on here, but essentially you can follow the colors. This individual, what we're looking at here are aerial views of the area of atrophy, and we've colored them in to make them easy to see. And so in May of 2017, this individual had an atrophy that was traced and here colored in orange. And then 1 year later, the area in red is larger. So that's just showing you that the area of atrophy grew steadily in the year before the patient received treatment. They received treatment right after the red measurements, and then we could see at month 9 in blue that the area of atrophy had gotten smaller. The green, you can't even see, it's totally covered, and then the yellow is at 23 months, let's call it 2 years, and it still remains smaller. This is very exciting to us. This patient also has enjoyed improved visual acuity, durable and consistent improved visual acuity throughout the 2 years. This is really an incredible outcome for us, and we're very excited and looking to reproduce this through some of the modifications that we made to our surgical procedures. And we dosed a bunch of patients this fall, and we're looking to see if any of those are going to also have this retinal restoration phenomenon. It's important because we think that it differentiates us compared to others' traditional approaches in the space. And I'm going to go through all of these companies, but suffice it to say that none of them has been able to show retinal restoration. In fact, I don't think any of them even believe that it's possible with their approaches, whereas we have actually shown it and had 3 experts in retinal imaging review the data and affirm to us that what we are reporting is in fact the actual event that's happened in the patient. We don't just rely on our cells, we also have improved the way that those cells are delivered without a lot of detail. Instead of going through the front of the eye and puncturing the retina and delivering cells, we've gained access to a new device which goes around the outside of the eye and comes up underneath the retina, and we're seeing better safety profiles with one of the main adverse events by changing to this new device. So we think our cells are critical. We think retina cells, not undifferentiated cells, are critical, and we also think using this device is going to add some important competitive advantages. So let's assume that we're going to be successful in treating dry AMD and provide some clinical benefits to patients. How are we going to satisfy an enormous market opportunity? Well, we've accomplished some wonderful things in the manufacturing. Our cells, our preparations are just thaw and inject. I mean, they come out of a frozen state. 5 minutes later, they're warmed up and they're put into the injection needle. It's about a half-hour surgical procedure. The patient is awake. It's a one-time treatment, and we are manufacturing today today. This is not future scale. Today we are able to manufacture 5 billion cells in just a 3-liter bioreactor. So we could have a wall of bioreactors put together. We could use larger reactors, and we're able to generate 2,500 clinical courses from one of those little milk jugs there. So it really is setting us up to try to be successful with this new transplant approach. I won't go through all the regulatory advantages that we have and the commercial advantages, but we think we're on quite a compelling track to be able to demonstrate that cell therapy really is the most suitable, appropriate, and promising therapeutic approach in an area which has no alternative treatments today. We have taken this technology and moved it into adjacent areas. I'm going to speak briefly about spinal cord injuries. These can be incredibly expensive, and they can cause enormous change of quality of life to the individuals who are affected. Unlike someone who maybe smokes 3 or 4 packs a day for a lifetime and gets lung cancer, you kind of wonder if maybe you saw that coming. Nobody expects to have a spinal cord injury. Nobody expects to have an epidural that goes wrong, leading to paralysis, or to dive accidentally into shallow water, and just the way that the wave goes ends up damaging them and causing them paralysis in all 4 limbs. This is a gentleman who suffered from an accident, a car accident, one of the leading causes. He was ejected from his vehicle, and he was paralyzed. He participated in our clinical trial, and of course you can see him now throwing a baseball out of this baseball game. He can operate his wheelchair. We've had a number of patients, about a third of our patients have had significant improvements, and that is very exciting. So let me describe to you what we're trying to do. We're trying to replace the cells that are lost following an injury, and we're targeting the cervical nerves that are high in the neck, especially the ones that are responsible for upper extremity locomotion. We want to be able to get your wrist and hand and finger mobility back. If we can recover that, that's going to drive a lot of mobility. Yes, walking is important. Yes, bladder control is important, but some of the injuries are so severe, it's really a bit ambitious to think that cell therapy could do that. But if we can improve an area of just a centimeter or so, and you can get back a couple motor levels, that would be quite remarkable, because this is what it means. I mean, here's exactly an example of the kinds of patients we're targeting. In the blue circle there, the C4, that would be a cervical injury at the 4th, let's call it nerve group, and it's responsible for certain upper extremity mobility. So if you have an injury at that level, you lose the ability to dress yourself, to feed yourself, to hold on to your coffee cup. And if we can move that level of injury down, if we can effectively heal the area and turn a C4 patient into a C6 patient, you can see massive improvements. Instead of around-the-clock care, now you're talking about people who just have a little bit of support around the home to help them with things like getting out to the store and so forth. So these are pretty significant changes, and we have seen these changes from our treatment. So how do we do this? Well, we obviously don't use retina cells. We manufacture something called oligodendrocytes. These are the cells of your spinal cord, which help support and myelinate neurons. So it's not enough to just have neurons grow in the area. These neurons are very complicated, and they talk to each other, and they need to be myelinated. So they need the material which insulates them and helps carry the signal from your brain to your fingers. And in doing so, by remyelinating those axons, you can reinforce their connectivity. So when they are wired together, they can then fire together. So your mobility is driven by a reinforcement of the activity. And so by providing the material or the substrate, the cells in particular, which are responsible for driving this activity, we are able to evaluate these patients and see if we can drive some improved benefits. Here's an image of what it looks like on the control versus a treated. This is a slice from an animal study, although, of course, again, we're in humans right now. But the difference here between untreated and treated is providing that material from which the patient can hope to regain some activity. I won't go through the design of the clinical trial for time, but we've got 25 people who have been treated. And what's notable is that all but one had a successful engraftment. We had no complications related to the cells. We can check them by MRI. And a third of the patient gained 2 or more motor levels. So remember that for just one slide. A third of the patients gained 2 or more motor levels. We've moved patients from C4 to C6. We've moved patients from C5 to C7. it is really exciting to think that people who maybe were going to have very poor outcomes were actually able to regain some locomotion thanks to our regenerative and restorative approaches. I won't go through other than to say that we have done some work to improve the manufacturing of these cells. Huge increases in scale, the functional assays. For this audience, really the takeaway is that there's been a maturation in the field. The field of cell therapy, the tools that we have today are so much farther ahead. So what the promises of a decade ago are now becoming clinical reality thanks to a maturation and advancement in the field. And we are very much trying to be a part of and lead that advancement. And this is one of my favorite slides, actually. The gray bars are measures of undesirable cells. So those are essentially impurities. And this is from the original material that was first used in the clinic. The orange bars, which are much lower, are measurements of our material, the material that we manufacture now. And you can see how much cleaner and purer it is. This to me is really a nice pointed way of showing that the technology that we apply and the cleanliness and reproducibility and scale and all the things that FDA cares about and patients should care about, those are things that we're able to do today, which were not possible a decade ago. So we're going to be thinking about how we can move this back into the clinic now that we've fixed the manufacturing problems, and we want to get this therapy to spinal cord patients. The last one, with just a fewer number of slides, is what we're doing in cancer. And down at the bottom here, this image, we're again expanding cells. We differentiate them into dendritic cells. Dendritic cells are so powerful. These are the cells that present foreign material to your immune system. So a lot of us have learned about coronaviruses and infectious diseases and vaccines, and that's what vaccines do, right? You're presenting foreign material to boost the immune system. Well, what if you could take foreign material, whether it's from a tumor or from a virus, and what if you could present it on 60 million dendritic cells and then inject those cells into a patient? We think that the patient's immune system is going to respond to that. And in fact, we've seen very high levels of reactivity to our treatment. And this provides us so many opportunities to develop new programs. The different antigens that we could use for different tumor types essentially allow us to design and map out dozens of potential programs. And those opportunities to partner with different companies around different antigens and different tumor types is really exciting. And we look forward to continuing to work with Cancer Research UK and move this program forward. So we'll have more to say about this during 2021, and we do have a clinical trial right now, which we hope to wrap up in the coming couple of months and be able to announce data from that oncology study. I'm not going to go through the news because I took too long to get my slides up, but just suffice it to say that we continue to crank out really interesting information using cell therapy. So Lineage is an exciting company. The company is well funded deep into next year. We have an enormous patent portfolio, and we really are an emerging leader. I want to save time for some questions, but if you want to learn more about what we're doing, we have some patient stories and some really amazing podcasts and other media type material on our website that I invite you to consider. Thanks so much, and I will stop sharing, and hopefully we've got time for some questions.
Great, fantastic. Thank you so much, Brian, for your presentation. So, questions. We have a question from Matt here. He says, very interesting, but the FDA seems skeptical about the possibility of cancer from stem cells. I mean, what do you think about that, Brian?
I think I'd need to know a little bit more about the question. Stem cells, so undifferentiated stem cells. Yeah, I'm skeptical too. I'm not sure, how long it's going to take to figure out where to use undifferentiated stem cells. There were a couple of sort of high-profile failures during 2020 with that. We don't endorse that. We use differentiated cells. Dendritic cells in particular for cancer have proven, I mean, there's an approved agent today which sells hundreds of millions of dollars that is based on presenting a tumor antigen by a dendritic cell to the body. The problem with that therapy is they're starting with the patient's own dendritic cells. So it's really like a custom one-off approach. We are using off-the-shelf approaches. So I think that there's definitely a lot of signs and evolutions from FDA that they are adjusting to the new reality around cell and gene therapy. And I think that is good for the field. But specific cell therapy. I mean, I don't think you can argue that CAR T has been revolutionary. The whole field of immuno-oncology has been revolutionary. We have a bunch of incredibly successful cancer therapies. NK cells, a different cell type, are really catching on. Monocytes, and then I think dendritic cells are going to be part of that as well. So I agree to the extent that you may be referring to undifferentiated cells. I disagree to the extent you're referring to specific cell types which have discrete functions in the body.
Great, thank you. Thank you so much for that answer. And Matt, I hope that answers your question. I'm sure if you have any follow-up questions, Brian will be able to answer them. So Greg has said, thanks so much for a great presentation, Brian. With all the changes we know occur with age, such as increased inflammation and ECM stiffening, do you think these implanted cells face a difficult microenvironment in order to survive and function properly in long term? If so, do you have any strategies to counteract this, or are you maybe thinking about it down the road?
Yeah, I do. I think, but I think that's also the point, right? We know that the microenvironment in the retina is unfavorable for the long-term health of the cells. These are cells that are old. They've been working very hard, helping you see things. But we're coming in with essentially, it's not necessarily correct to think of it as a younger cell, but it's a cell that has all of the vitality and vigor that you would want. And so it's sort of like converting, this is kind of a silly way, but I think of it as planting young saplings in a fallow field, right? You're actually driving the balance backward by putting these vigorous retina cells into an environment and asking them to help everybody in the neighborhood do a little bit better. So when your neighbor, puts an addition on their house and the home values go up in the area, right? These are silly analogies, but conceptually that is part of what we're doing. But your question about how long they would last, I mean, I really think they're going to outlast the host, so to speak. I mean, these engraftments have remained durable. This is an interesting story. We initially began with a year-long of systemic immunosuppression. So we wanted to immunosuppress people so they wouldn't reject the cells. And then that was for the very first patients. Then we peeled back from a year to down around 3 months. It became physician's choice, and they were taking patients off the low-dose tacrolimus for about 3 months. We recently treated someone with no tacrolimus, and we haven't announced how they're doing yet, but you could obviously see the direction that we're headed in. We haven't had problems with rejection. We haven't had to go in at 6 months or 1 year or 3 years and redose. So I think at least in the eye, which has a bit of immune privilege, in the spinal cord where you don't have rejection, I think we're going to see that we can tolerate these cells longer than we can live. But why not just put in another dose? If you outlive your new ones, you can put in another replacement part.
Great, thank you, Brian. I hope that answers your question, Greg. Beverly has asked, really exciting presentation. She was wondering if the starting cell line is the same for all of your products. How has this influenced your regulatory approvals?
Yeah, so they're not. So cells are funny. Some of them like to become neural lineage cells, and others do not. There are many, many, many cell lines available to work with. I think even just in this country, there are about 400 different cell lines that are on the NIH approved list for use and to receive federal funding. So we use one of the oldest lines, and it's been around for about 25 years, so it's incredibly well characterized. And we actually perform characterizations and analyses on our cell lines that go far beyond what FDA is asking from companies today. So, for example, for our retina cells, it's one thing to karyotype and just count chromosomes, but that's sort of like the most simplistic thing we do. We are looking to ensure that they have activity so that they can phagocytose material. We make sure that they can polarize. We want to make sure that they know how to orient when they get into the retinal space, because these cells they basically have an up and a down, and they secrete different things based on which side of the cell you're looking at. So they need to orient correctly. We look at a large number of surface markers. Those are our indicators that we're looking at retinal cells and not epithelial cells or islet cells or whatever. So we have a very long list of in-process controls to help ensure that we are directing the lineage the right way, and then release criteria before we release clinical material that's going to go into a human. we make sure that it meets all of these criteria. As I say, what we're doing is far beyond what's asked for from the FDA. I think the FDA is going to increase their criteria over time, but for us, it's just we just happen to have a higher level of quality control than the minimum which is required, and I think that's important for patient safety.
Great. Thank you, Brian. I hope that answers your question, Beverly. I see here Pavel's got a question. How do the costs of cell therapies compare to other therapies? But due to time, Brian, I hope you can answer that on the chat for Pavel. But yeah, thank you so much for your time, for your presentation, answering our questions. Yeah, and if anyone has any follow-up questions, please do ask them via People's tab. But thank you so much, Brian.
It's been my pleasure, Hu. Thank you so much. Bye bye.